Recording in progress. I'm converting my, okay, close that. Switch to correct the problem. Trying to make a PDF, but I'm having an issue with this. Cancel, retry. See if that does it. Okay. We're gonna do it. Did we? It's looking like it. And then I can upload that to our Canvas page. Hey, everyone. Close that. Talking about what we're doing. Okay, so today what we're going to be doing is we're going to be introducing a pretty important topic, and that is enzymes. Um, some of y'all might be familiar with what an enzyme is, and maybe you've heard the term before. Maybe you've heard it outside of, biochemistry class. Maybe you've heard it in the context of like digestive enzymes or something like that. I think that's kind of a popular way in which to hear that term. Um, where is my file? Um, and as sort of a primer for that, I've got some poll questions for you guys right off the bat. I'm gonna leave these up for about 5 minutes. Um, just as a fair warning, my computer wasn't screwing up with its audio. However, it was running noxiously slow earlier this afternoon, but I'm hoping that's not going to be an issue today. Though right now it's not really giving me much confidence. Okay, so here we go. Here is your enzyme or your questions. Then going to drag it to my PDF. It's good. Drag over my PDF. Click drag. Submit. Take a second to upload that file. Okay, we're good. Save. All right. And then what I'm going to do is exactly what I did last time, where I just open up the slide deck and don't go full screen on it, because then that's where real problems begin. Okay. So I'm doing a little bit of trying something new when it comes to introducing this topic. Um, because I've tried, traditionally you kind of approach proteins as kind of one topic, and then you approach enzymes as a separate topic, you know, but there's sort of a hierarchy to it. You start with proteins and then you go on to discuss enzymes. So I'm going to try to approach them simultaneously. And the reason for that is because there's lots of these things just kind of are threaded together. So I want to try to, oh, sorry, enable. Um, I want to try to avoid some confusion because there's lots of overlap. And whenever we approach that or see that overlap, I want you guys to look at it and say, like, hey, that doesn't make sense, or hey, that seems like that's redundant. Because there are definitely some things that are redundant in the conversation about proteins and enzymes. So the last thing that I want to cover regarding, or one of the last things that I want to kind of tackle is I want to tackle the conversation of protein structure. We've talked about protein structure and we've talked about primary structure, secondary structure. And then just the other day on Monday, I talked about um, your tertiary and quaternary structure. Um, if you remember, your tertiary structure is just the 3D structure of a single polypeptide. So tertiary is the 3D structure of one peptide. And I'm just going to say peptide, but remember that we're talking about something that's typically more than 50 amino acids. Your quaternary structure is going to be kind of the same thing, but with multiple So I'm going to just go kind of quotation there, multiple peptides. And both of these really are stabilized and revolve around R group interactions. Um, so that's why I spend a lot of time and think that it's very important that you're comfortable with your R groups and know what they look like. So we can go from there. So tertiary structure, the folding of a polypeptide chain in three-dimensional space is referred to as the tertiary structure. This is concerned with the arrangement in space of all atoms in a polypeptide chain. The conformation of the side chains and prosthetic groups are included as well as the packing of the helices and sheets. Now, this term right here, prosthetic groups, that's a term that is, I think, valuable to know. And we're going to see kind of the application of one of those prosthetic groups in just a moment. Now, prosthetic groups, I always think of a prosthetic group of a protein as similar to how I think about a prosthesis that a person may have, whether that's a prosthetic foot or prosthetic hand or something. Essentially, a prosthetic group, the way that I think about it is it helps, in this case, a protein carry out a function, carry out a function it otherwise wouldn't be able to do. I'll say would struggle to do. And in this case, whenever I mean to carry out a function, what I'm meaning is, well, this function would be more than likely, I'm gonna go with kind of lay terminology here, do some type of chemistry, type or sort of chemistry. So what that means is, well, maybe the amino acids that make up that protein would not be able to bind a particular molecule, but this prosthetic group assists and makes it capable of doing that. So um, the formation of the tertiary structure is primarily determined by the interactions of the amino acid side chains with each other and the backbone atoms. So, your R group interactions, like I said up here, well, those R groups might also interact with the backbone. Now, I want you to keep in mind secondary structure. Well, the secondary structure is stabilized by H bonding of the backbone. E-O-N-E. But more specifically, backbone atoms with backbone atoms. So this is where we have a difference between the secondary and tertiary structure. It's backbone atoms interacting with backbone atoms for the secondary structure, whereas tertiary would be like, well, backbone plus R groups. Okay. Um, and then quaternary structure, well, again, this is all about different or multiple peptides. So backbone BB of peptide 1 interacts with R group on peptide 2. Okay, so could you guys give me on a scale of 1 to 3? 1, I feel really good about my understanding of the primary through quaternary structure, and a 3 being I feel good about, primary and secondary, but I'm struggling with tertiary and quaternary. And I didn't really give a spectrum there, but yeah, you guys got the point. Okay, cool. So I'm seeing lots of ones. I'm happy to see that. We're going to look at some applications of these different ones throughout the semester, specifically the tertiary structure, how the R groups can interact with the backbone. And whenever we see that, I will bring that back up just to emphasize that point. Okay, so with respect to the tertiary structure, I want you to know this definition, know that we're talking about R groups interacting, We've also got to kind of grapple with or wrap our minds around these prosthetic groups, which we're going to see one today. But then I also want you to really take this point home and think about different amino acid R groups and how they might interact or not interact with one another. For example, I would, do my best to kind of come up with some like right off the cuff, hey, you need an R group interaction for a tertiary structure. I gotcha. A serine and a threonine. Those two amino acids are going to interact with one another through hydrogen bonding. Arginine and let's say aspartic acid, well, those are going to have an ionic interaction. So think about those and just kind of have those ready to go. Now, with that said, this last little detail, we talked about the Chow-Fastman prediction model and that scoring system that we had for different amino acids in the secondary structure. So you can predict a secondary structure. Tertiary structure is much more difficult, and it has remained, or sorry, it's easier now than it was 5 years ago, but it's still a very complicated process. Um, and that's just because of all of the different types of interactions, all of the different folds that a protein can take. So tertiary and quaternary structure is still fairly complicated, but we have much better models or prediction models now than we did, 5 years ago. And being able to predict a tertiary structure, you might be wondering why. Well, a good reason for why you'd want to be able to do this is because, sorry, um, drug design. If you can predict what a protein looks like simply based on its amino acid sequence, then you don't have to go through the process of expressing that protein, purifying that protein. You can make lots of predictions before you go through all of that. And of course, you probably want to validate it with bench work and experimentation where you see things, but drug design is a great reason why you'd want to know tertiary and quaternary structure. Now, the quaternary structure of a protein, many proteins consist of more than one polypeptide chain. These are going to be known as subunits. These are your different polypeptide chains. The individual subunits associate in a specific geometry for that protein known as the quaternary structure. So I just want you to keep that in mind as opposed to like when one protein's floating around and it bumps into another one, that doesn't magically become a quaternary structure. There's a little bit more stability to it. Now, these subunits are going to interact via or through non-covalent interactions, and these proteins with more than one subunit are typically going to be known as oligomers or oligomers. Your dimers, trimers, tetramers, etc. Now, proteins can vary tremendously in their complexity and their composition. For example, the largest known protein is a protein known as titin. It is a connective tissue protein, a muscle tissue protein. One titin is approximately 34,350 amino acids long. It has a molecular weight of 3.816 188 Daltons. If you remember, one amino acid, one AA, on average, is about 107 to 110 amino acid Daltons. Sorry. Now, to put into perspective, as a graduate student, I worked on a protein that was 765 amino acids, and that was considered kind of a difficult and large protein to work with. So when you look at this spectrum here, pyruvate decarboxylase, that's a metabolic enzyme that's 2,252 amino acids long, but that is made up of four different subunits. So when it comes to the complexity of proteins, they vary tremendously. In addition to that, it's not exactly as simple as saying, oh, well, a small protein is an easy protein to work with because the environmental conditions vary quite a bit. Small proteins might be associated with a membrane. Membranes are going to be super hydrophobic, so that makes them difficult to kind of navigate through because, well, they're going to be hydrophobic, they're going to hate water, so purifying them is going to be difficult. Now, Do I want you to know all of these numbers or anything like that? No, I don't. What I want you to understand and appreciate is that proteins vary pretty dramatically in terms of their size. And there's not really a golden rule of, well, if a protein is more than 150 amino acids, it's multiple subunits because we've got Titin down here. That's one subunit. That is a monomer, excuse me, and it's 34,000 amino acids long. Now, a kind of classical grouping of proteins is to first and foremost classify them as globular and fibrous. Your globular proteins, more or less, or arguably, are the ones that are kind of, not to say, not to give a golden rule if this is always the case, but if you were to pursue graduate studies and characterize an enzyme or characterize a protein, chances are you're going to be working with a globular protein. So these are maybe a little bit more common for study and for analysis. Now, your fibrous proteins are proteins that you've probably heard a little bit more about, or maybe you're familiar with the names of fibrous proteins, but might not know them as fibrous proteins, or you do. Collagen is an example of a fibrous protein. Your fibrous proteins, they contain polypeptide chains organized approximately parallel along a single axis. So to kind of sum that up, well, they have structure, like a clear structure, structural rigidity. Say that. Now, in addition to that, these typically, their primary structure follows a pattern. I'll say follows a bit more of a pattern. Now, with that said, I'm saying this uniquely about fibrous proteins because globular proteins, their primary structure, no consistent and obvious pattern. Consistent and obvious pattern. Obvious pattern. Okay, there we go. Globular in terms of well, what about their shape? What it's a glob. Yeah, exactly. It's kind of an amorphous shape. I would say more times than not, it's semi-spherical. Um, but these proteins can become much more complicated, and their shape becomes a little bit more abstract when you start talking about like a tetramer or a 26 mer. And I'm saying 26 mer because I don't know what the prefix would be for the number 26. It'd probably be something like if deca is 10, dodeca I think is 12. I'm going to stop there because I don't know beyond that. And if dodeca is not 12, then so be it. Okay, so when it comes to proteins and their structure, what I want you to know is, and what we're going to be getting into a little bit more, is that tertiary and quaternary structure area. We're going to primarily, for a little bit, be focusing on globular proteins. And I'm going to use some examples of globular proteins. Now, today is, it's, I want you to think about today as like, okay, we're talking about proteins, and now we're going to classify them. Okay. And here's one classification, here's another classification, and there's some overlap in the Venn diagram of those two classifications. So right now, what we're going to pivot to is functional proteins. So we're not really talking about the shape, but there is like a shade of this conversation that has to do with their shape. Functional proteins, I would say more times than not, more than not, are, N-O-T, are globular. Now, these functional proteins, they carry out a function. In this case, whenever I say function, I'm not referring to catalyzing or doing a specific type of chemistry, because these functional proteins that we're going to go through here are not going to break any bonds. Now, when it comes to functional proteins, we want to introduce this new term right here, ligand. And I remember as a graduate and undergraduate student, some people would, I'd be in a class one semester and someone would say protein ligand binding. And then the next semester, somebody would say protein ligand binding. And I would say to myself, I don't know if either of those are right. Not that there's a third correct one, but it seems like the sort of debate I don't really want to get in the middle of. So the function of many proteins involves the reversible binding of other molecules. So pick it up, release it, release it. Now, that other molecule is going to be referred to as a ligand. So I want you to think about if something can be bound and released, that thing that is bound and released is a ligand. I will say ligand. I'm sure that's, I don't know. I don't know. Ligands can be a variety of different molecules. I think it's pretty common to think about a ligand as being something small, maybe something like a sugar or something like that. Sugar. But don't get it twisted because that could be a protein. It could be a small organic molecule like a sugar, or it could be a inorganic compound. So it could be something like, an aluminum complex. Could be a number of different things. Now, the key about ligand binding and what defines a ligand basically is the protein that binds a ligand is first and foremost non-catalytic. So the binding and release doesn't result in any sort of reaction taking place. Now, this often causes a protein conformational change. This term right here, I don't think I've mentioned that. If you've heard it before, fantastic. If you haven't, don't worry about it. Conformational change of a protein is a really it's a $5 word way of saying shape change. So there's a couple of words, a protein shape. Sometimes you'll hear referred to as a fold. And you'll hear something like when a protein achieves its native state, that would be its fold, that would be a conformation. So this conformational change is where a protein goes from one shape, one state, to a different shape or state. Now, in addition to that, this ligand binding can indeed be regulatory. So it can stop a process or it can accelerate a process. Um, a good example of this and a good model for this is in the transport of oxygen, because the oxygen, the O2 that your body takes into your lungs, is deposited as O2 in your muscle tissues. This O2 molecule that's picked up in your lungs and deposited in your tissues is unchanged throughout this process. So we have a vehicle or a protein that does all of this. The protein that does this by transporting it from the lungs to your tissues is known as hemoglobin. Now, oxyhemoglobin is the form of hemoglobin that has oxygens. Now, it's difficult to see this, but what I want you to see is this right here. This kind of looks like a Trivial Pursuit piece with the four different FEs is showing the one, two, three, four O2 molecules that hemoglobin can bind. So hemoglobin, shorthand of HB, can bind four O2s simultaneously. When that hemoglobin is fully saturated with O2, fully oxygenated, it will transport those four O2s and deposit them in the tissues. And in the tissues, you'll find this other protein known as myoglobin. Myoglobin will pick up O2 and hold on to it until it's needed, in which case it will release it. And that O2 will find its way into this right here, which I know that there's at least one person. And give me an I in the chat if whenever I pointed to this structure, you said the powerhouse of the cell. That's the mitochondria. It's just muscle memory, and I'm happy about that. So that mitochondria, what it's going to do is it's going to help break down molecules like sugars and fats and extract a bunch of energy in an oxygen dependent process. Okay. Now this oxygen that's deposited here will generate CO2. Myoglobin stays in the tissues. Hemoglobin, remember it brought O2 through the arteries into the tissues or by way of the lungs, or no, from the lungs by way of the arteries. Oxygen was brought by hemoglobin, deposited in the tissues. And as it turns out, hemoglobin also functions in a little bit of an expiration process by transporting CO2, which is a waste product, through the veins into the lungs where it can be exhaled and released. Hemoglobin basically stays in the bloodstream and just cycles back and forth, going to tissue to drop off O2, pick up some CO2, go back to the lungs where it can release that CO2. So all the while, hemoglobin and myoglobin, which is denoted as MB, they've done nothing to break any sort of bonds. They've held on to O2 whenever they needed to, and then they released it whenever the cell said, hey, you better release that. Now, as a comparison between myoglobin and hemoglobin, these two proteins function in oxygen storage. Myoglobin, or MB is how I will denote it, functions as an oxygen storage and transport protein in muscles. Now, this transport right here for myoglobin, it's not long distance. It's basically within muscle tissue and within cells, holding onto it, releasing it at the mitochondria. Hemoglobin functions in an identical capacity in the bloodstream, in blood vessels. Now, myoglobin and hemoglobin are down here. This is myoglobin or MB, and this over here is hemoglobin or HB. These two proteins are built on a common structural motif. This structural motif is known as a globin fold. Now, if you're hearing this term structural motif for the first time, you're not alone. Structural motif, you could kind of think about it as a protein's blueprint. And what I mean by that is, think about a house blueprint. Okay, we want a door here, we want a fan here, we want a bathroom here. Well, protein or a protein's blueprint, and when it comes to a structural motif, it would be okay, we want an alpha helix here. And then after that alpha helix, we're going to have a what's known as a beta turn. And then after that beta turn, we're going to have another alpha helix. And it just kind of maps it out, this specific kind of structure, as opposed to something like alpha helix, alpha helix, alpha helix, alpha helix. So there's an organization to these secondary structures. Could you give me a 9 in the chat if you feel all right about the term structural motif? And any other character if I'm a little mixed up on them. Dang, it's a lot of 9s. Okay, so hemoglobin and myoglobin, if you look at these two structures, they're very similar to one another. Now, the polypeptide chains of myoglobin and hemoglobin fold around a prosthetic group. There's that term, a prosthetic group, and this specific prosthetic group is known as heme. So heme is a molecule that gives this polypeptide and these collective polypeptides a function that they would otherwise are less capable of doing. Now, that prosthetic group basically, gives a function, gives a new function to this protein. Now, I want to take a step back to kind of clear up some things about the structure here. And actually, no, I'm going to go ahead and jump forward to this slide. Here we've got myoglobin or MB highlighted. Now, what's difficult to see here is this right here is referencing the N termini of the polypeptide. This references the C termini. Okay, so this is a polypeptide of, I think myoglobin is something like 110 amino acids. and all of the different hemoglobin subunits are somewhere between 105 and 120 amino acids long. So we've got our N termini, and then this letter A right here, B, C, D, E, F, G, and H. These are all referencing different alpha helices. There's the A alpha helix. There's the B, the C, the D, the E, the F alpha helix. Now, if you compare that to your structure of hemoglobin, and let's take this, what is identified as the beta 2 subunit, because that one is the only one that has the N termini right here and the C termini represented or clearly denoted. If you look at all of these, A, B, C, D, E, F, G, and H, the same alpha helices. If we were to take this myoglobin, rotate it, and overlay it over this subunit, we're probably going to get a whole lot of overlap in terms of the different movements of that ribbon or of that polypeptide. Now, that illustrates the similarity, and I hope you guys see that, but a fair question is why? Why is it labeled the A alpha helix? Why the B alpha helix? And in terms of, if your question is why is it the A alpha helix as opposed to alpha helix number one, I can't answer that question. But for the A, B, C, D, and so on, this is helpful because hemoglobin and myoglobin have both been very well studied. And it gives a way of identifying significant and important amino acids in the function of this protein. So what you might say is, um, let's say A residue number 6. So A6, what that is, and that looks like a lowercase b. There we go, there's a 6. This would be a way of communicating, hey, the 6th amino acid in alpha helix A is important for binding of O2. And to kind of bring that point home, could you give me a W in the chat if you have ever heard of sickle cell anemia, and an O in the chat if you've never heard of sickle cell anemia? Okay, so I'm seeing lots of W's. Sickle cell anemia, as it turns out, I'll say SSA, is attributed to one specific amino acid mutation. Okay, so if I remember correctly, it's a aspartic acid mutated to a valine. So we've got a residue that can be involved in ionic bonding being mutated to something that's a non-polar residue. Now, I mentioned sickle cell anemia because it's an example, but that's not the only hemoglobin mutation. There's lots and lots of different hemoglobin mutations. And those mutations kind of get this language of, well, the C helix residue number 7. So it's a C7 mutation. And so it's all about kind of geography of the protein and giving some more specific identifiers for what amino acids cause this functional difference. So that's why we use this A, B, C, D, E, F, G, and H to identify these different helices. All right. Now, I've already kind of shown you this, or you've probably been able to deduce this, that hemoglobin is made up of 4 subunits: alpha 2, alpha 1, beta 2, and beta 1. So 4 subunits. The composition of hemoglobin we'll get into a little bit more and how those different subunits interact with one another later. But for the time being, what I want you to know is that myoglobin is a monomer, Hemoglobin is a tetramer. Myoglobin as a monomer binds one O2 molecule. Hemoglobin binds four O2 molecules because they all have this guy right here, heme, 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 heme. All right. Now, to kind of illustrate that point, oh, I got a private message that says you don't have to answer this now. Well, now I'm distracted. I got to copy and paste that into it's a longer question. So I'm going to copy and paste it into a Word doc so then I can revisit it. And that's why part of your screen is obstructed with a gray box in the middle of it. But uh-oh, I'm asking my computer to open two programs at once. I'm playing with fire, y'all. Okay, there we go. All right, so now I'll minimize that and keep my fingers crossed. Okay, so now let's look at myoglobin. Here, what I want you to recognize is we've got, I like this model because it kind of gives us like um, it's almost like a structural model or a skeletal model of our protein, of our myoglobin protein. And we've got helix A, helix B, and then we got this AB. Well, what's AB? AB is just going to be a term. So it's a it's a transition in the structure where this is a beta turn AB, connecting alpha helix A and alpha helix B. Now, 90% of the protein is an alpha helix, divided up amongst all of these different alpha helices. Now, what I want to draw your attention to right here in the middle with this, let's call that magenta. I'm sure somebody's better with colors than I am, but that magenta, no. Petra's, no, straight up, no. Not even magenta. I don't know. Cerulean. Okay, so we got that pinkish colored molecule in the center. Hot pink. Oh yeah, I suppose that works also. Okay, so we've got this hot pink circle in the middle of the structure. I know it's not cerulean. I just love that word. That is representative of our iron ion. So in the center, this structure right here, this ring structure, is our heme molecule. In the center of it is an Fe2 plus ion. That's what we want to be in the center of it. Why? Well, O2 or oxygen is not very soluble in an aqueous environment or an aqueous solution. So O2, you can't just hope that it goes into the solution, hope that it goes into your blood. You have to have something else that assists with that. Its solubility. So O2's, I'm going to drop that and say O2's solubility and transportability is increased when it's bound to a soluble carrier, like this iron ion. Now, amino acid side chains are not going to be able to bind O2 very well, regardless of the amino acid. So, like, if you think about if you think about the functionality of different amino acid R groups or the appearance of those different amino acid R groups, like you've got some that are good at hydrogen bonding, you got some that are good at ionic bonding. But to bind an O2 molecule, they're not very good at that. However, transition metals like Cu or copper and Fe or Fe or iron, they're very effective at that. Now, those transition metals as ion, Fe2 plus specifically is a good one. So iron is incorporated into a prosthetic group, heme, and it can be used to transport O2. Now, if anyone has ever decided, or if anyone is a vegetarian, there's always the person who's their friend who's like, I remember I did this one time where I was like, I'm not going to eat meat anymore. And immediately all of my friends were like, hey, Joel, we're going to be super supportive of you. Not really. Instead, what they were was like, well, that's dumb. And I was like, thanks, friends. And they said, you're not going to get enough iron in your blood. And I was like, okay, well, that's a tale as old as time. Thanks for the support, guys. Yeah. So Um, anyway, friends are the best. They are, but sometimes they can be rascals. So the point is that this heme structure right here and this iron in the middle of it is an important thing to have as a part of your diet because that helps transport oxygen. And if you don't have enough iron, which um, you know, steak is always the classic example of something that has a lot of, or beefsteak is an example of something that has a lot of iron. So that's always the, well, that's what you need to eat. You need steak. There are other sources of iron, but it's very biologically available and accessible from a meat like a steak or something like that. But the point is, we need to have this iron as a part of our diet to help make O2 soluble and to help make O2 be able to go into our lungs and perfuse throughout our body. Now, I've got a poll question because it's relevant. I'm going to throw that poll question up. I'm going to take a drink of my soft drink. There we go. Now, with respect to this iron ion in the center of that heme ring, there are amino acids that help out with this. For instance, this down here, this is a histidine ring that basically helps to hold this iron in the center of that heme ring. So when it comes to a prosthetic group helping, yeah, absolutely. A prosthetic group helps to position this iron so that it can bind O2. Now, that is what is denoted also right here. This blue ball represents another histidine ring. So when it comes to coordinating that iron and coordinating subsequently the oxygen, there are actually two histidines, specifically a proximal histidine. The proximal histidine is directly coordinated with the heme ring. So it is right underneath the iron within the center of the heme ring. And there is a distal histidine. This is not directly coordinated with the heme ring. So Fe2, which remember a Roman numeral, that means 2 plus ion, is coordinated by the 4 porphyrin N atoms and 1 N from histidine F8. So I'm going to jump back just a second. This is our heme molecule. Now I'm going to circle those four, or actually, I'm going to highlighter crashed my computer earlier, but here we go. Those 1, 2, 3, 4 porphyrin rings. Didn't crash it this time. So we've got that iron that's coordinated by those 4 rings as well as this ring of this histidine. Now, those rings help hold that iron in place. Go ahead and end the poll. My question that I posed for you guys was how many O2 molecules can myoglobin bind? And with 73% of the vote, 1 was selected, and that is the popular answer. It is the correct answer. So myoglobin can bind 1 O2 molecule, hemoglobin can bind 4. Keep those two numbers straight, and you're going to be in good shape. All right, next up, we got to get my other poll question, see where we are with that. Now, when it comes to, well, what amino acid is responsible for this? This image and this depiction right here is a really good one. Now, what I want you to be kind of thoughtful of is this model shows, I've got another poll question up. Go ahead and answer that. The poll question reads, which amino acid is responsible for coordinating the O2 binding to heme? This model right here shows the two histidines that are responsible for binding O2 in the center of the heme. And this also shows a valine and a phenylalanine. And you might think, oh, well, those must also be important. And I think that using the word important, yeah, they're important. But these two residues, valine E11, which one thing I want you to recognize is that is referencing amino acid number 11 in the E helix. Remember the A, B, C, D, E, and so on. So it is important, but it's not mission critical. This phenylalanine, this is amino acid number 1 in the C, D turn. Is it important? Yes, it is. Is it mission critical? No, it's not. Compared to the histidine E7 and histidine F8, the phenylalanine and the valine are really not quite as important. Okay, I'm going to go ahead and end the poll. We got another midterm question or midterm poll question popping up in a little while. I'll share that with you. But the popular answer was the correct answer, H for histidine. Okay, now I'm gonna. Okay, so what I want you guys to know about proteins and their function is kind of captured with myoglobin and hemoglobin. There's a couple of different amino acids that are involved in coordinating, putting this iron ion in the right place so that it can bind O2. Now, we're going to change gears a little bit. And we're going to talk about enzymes. Now, enzymes are the biological agents of change. Now, our language and the terminology that we use for enzymes also changes. Now, this brings up a couple of important terms. This enzyme that we're looking at here is one known as lysozyme. Lysozyme is a good enzyme for breaking down sugars that make up the cell wall of E. coli. But this brings up active site, which is kind of that's the important region on an enzyme. And it's pretty common for people to say or use the term active site. So I'll just label AS residues. And what that means is people, if you ever hear someone say, oh, an active site residue, they are referencing amino acids, I'll just say AAs, in the active site. And more specifically, AAs that break a bond or break bonds. So active site residues are those residues that are involved in the chemistry to break a bond. Okay. Now, taking that back and applying that to our conversation about myoglobin and hemoglobin. Well, we talked about ligand or protein ligand binding. When we talk about an enzyme, we drop the term ligand and instead use the term substrate. So a substrate is something that is going to be acted on, and more than likely a bond is going to be broken. Some electrons are going to be pushed around. That is different from a ligand. A ligand will not be changed by the binding to a protein, whereas a substrate more than likely will be. Now, so substrate and ligand are similar, but different. When you hear substrate, think, oh, okay, going to be broken. And Ernest, I hope that answers your question. If not, please let me know what that question is. A substrate binding site is, well, just that. Where on the enzyme, what part of that protein, what part of that enzyme does the substrate actually bind to? Now, with that in mind, there's one kind of term or definition or one thing that I want you guys to be thoughtful of. When you hear enzyme, and actually I'm going to go up to the previous slide. An enzyme is a biological agent of change. Okay. I think for a very long time there was this misconception that all enzymes are proteins, but not all proteins are enzymes. So a takeaway from this would be, or this idea would be enzymes, or let's say proteins, are amino acids in a polymer or polymers of amino acids. Let me change that. Are polymers of amino acids. Okay. And then the same thing. Enzymes are polymers of amino acids as well. Now, there has in the past, I don't know, maybe in the late 90s, early 2000s, there was a little bit of a revelation that that's not exactly like this remains true. This definition remains true. But this is invalidated because of these compounds known as ribozymes. And these are enzymes made up of RNA. So, an RNA molecule or a class of RNA molecules with a catalytic function. So, an RNA molecule can make and break bonds. So, that's where this idea, enzymes are uniquely made up of amino acids, kind of fell apart. Now, with that in mind, for this class, we're going to be talking about enzymes using the kind of classical definition that most enzymes are just a bunch of amino acids, are a or are polymers of amino acids. But this detail right here, I want you to keep that in mind and be thoughtful of that because I don't want you to kind of be living your life willy-nilly thinking all enzymes are made up of amino acids. Now, one thing that I will do that I do need to work on is I will many times refer to an enzyme, specifically like lysozyme here. I'll use the term protein interchangeably when I'm talking about lysozyme. Now, lysozyme is a polymer of amino acids, so it's a protein. Carries out a function, though, so a better thing to say is to describe it as an enzyme. Now, I know that's always a little bit confusing when you say, well, this is true, and then the opposite is untrue, and I apologize for that confusion, but don't get too overwhelmed by that. Now, when it comes to an enzyme and a substrate binding, what I want you guys to learn about here in our kind of brief intro to enzymes go is there's a pattern. So there are some enzymes that have a single substrate and they will produce a single product. So you could see a schematic that looks like this. This straight line represents our enzyme. The substrate binds, and then enzyme does its work, and the product is released. I'm sorry to distract myself, but the way that the light is shining through my blinds makes it look like I have a light mustache. Okay, now as an alternative to this, there are enzymes that can have two substrates and they can have two products. Well, in this case, a single substrate, multiple products, we've got our enzyme on this straight line, we've got our substrate that binds, and then two separate products are released, B and C. Now, this notation right here with the straight line is something that I want you guys to be familiar with, kind of how to process that. Okay, if there's anything about that that doesn't make any sense, please don't hesitate to say as much or ask anything about that. So here again is our enzyme that can have two substrates, A and B. And our enzyme might function by binding A and B, consolidating them into one thing, and releasing our product, C. Our last model is the one that adds a little bit of complexity to it, to this kind of proposal. Our last model is our enzyme, represented by this straight line, has two substrates that it binds, A and B, and then two products that it releases, C and D. Okay. Now, before I proceed, not necessarily do you have this mastered, do you understand every aspect of this, but my first question, and give me a one if yet, or give me a Y or an N. Do you follow that our enzyme is represented by E and our products and substrates are represented by A, B, C, and D? Okay. Now, this last one is where we have a little bit of complexity to it. Now, I'm going to add another slide after this because I feel like that's the best way for me to do it. Okay. So when you look at this right here, enzyme, substrate A, substrate B, substrate C, substrate D. I'm guessing there's at least one of you out there who's looking at this and they're like, huh, is there a reason that you wrote A then B and then that you wrote C then D? Would it be, is it the same thing? And I'm changing the color, but is it the same thing if you said B, A, C, D? If basically what the difference between these is saying is that A and B are binding in different sequences. Are there certain enzymes that function where maybe A has to bind, then B can bind, or vice versa? Can everyone give me a one in the chat if they understand the differences between the models that I'm showing here, and a nine in the chat if that's confusing? Okay. So the idea is, well, are there enzymes that bind substrates in random orders or patterns? And then opposite of that, what about, are there enzymes that bind in, what about specific sequences? And the answer to both of those questions is yes. There are some enzymes that they do have to have a very restrictive pattern, which is where we get these different scenarios. So there are enzymes that follow an ordered pattern. The sequence of subs binding / product release is absolute. It must follow a specific pattern. And then random indicates the opposite. So I'll just put no. Does everyone give me a 7 in the chat if you understand what I mean whenever I say no in reference to random? I'm really contrasting that with the ordered system. Okay. Now this last one, I want to reserve that one because it's kind of complicated. So I'm going to clear this out. And I want to give you these models so that then I can give you examples. So a ping pong model. It's a little bit more complicated. I've got my straight line. Yeah, it's straight. It's just on an angle. And I've got my enzyme. My enzyme here is going to bind my first substrate A. And if you look at that line as being this straight line right here representing my enzyme, I'm going to go ahead and tell you that this will form a complex. My enzyme and my substrate will form a complex. That complex consists of E and A. Now, this enzyme will, you could think about it as it will take apart, so a portion of my substrate, and add it to itself. And that enzyme will become no longer the exact same enzyme. Instead, I'll label it as F. So E and F are different forms of the enzyme. And in that process, so E becomes F, A becomes something else. I'm going to say A becomes my first product, R. And I'm sorry for switching the letters around. I just want to throw some different ones at you so that it's like, okay, this is starkly different from what we saw previously. So that first product, R, is going to be released here. So in this ping pong model, what happens is our enzyme binds to our first substrate, A, and we get the EA complex. Some sort of reaction takes place here that changes our enzyme to be, well, F, a new version of our enzyme. Can you give me a 3 in the chat if you understand that E and A are the enzyme, but they're slightly different? I always like to think about it as like F is a methylated form of the enzyme. It just took a methyl group off of the substrate. Now, that demethylated substrate is our first product, or R. Okay. So our substrate has changed to make our new product, but now we're at a point where we have F. It's the enzyme, but it's different. And it's at this point in time where we would bind our other substrate, B. Okay. Now, F, our different form of our enzyme, will bind to B, forming this FB complex. Now, when F and B form this complex, some sort of reaction is going to take place. Say that methyl group that I alluded to, the methyl group from here on A that ended up on our enzyme to make our different form of our enzyme now gets transferred to our substrate. So it gets transferred from our new form of our enzyme to our second substrate. So what does that mean for our enzyme? F reverts back to E, our original enzyme form. So what that results in is our second substrate, B, gets modified. And let's say that that forms our second product, Q. Enzyme is then back into its original state. So what I'd like to do is ask, so what I'd like you to imagine is that here is our first substrate. Just that blue dot with a methyl group on it. Absolutely. So sorry, would you mind repeating again? Why does it go back to its original state? I'm on campus and my Wi-Fi keeps cutting out. If I miss a poll or something, that's why. Sorry for the inconvenience. Don't worry about it. Okay, so here's my enzyme. I always like my enzyme to be a Pac-Man. So this is E, this is A. Now, when these two things get together, that methyl group is going to be transferred from A onto E. So now this is no longer E, but is now F. This is no longer A, this is R. Can everyone give me an 8 in the chat if you understand E versus F and A versus R? Why does a go Tor? Because I just randomly selected R as a different letter, a different symbol. That's why R. So don't get trapped on like deciphering the specific letters. So now F, our Pac-Man with a methyl. Pac-Man with a methyl group can bind B, our second substrate. Now, what's critical about this is that this substrate here, our free enzyme, our original enzyme, our as one of our classmates said, our OG enzyme cannot bind B. E cannot bind to B. Only F binds B. Now, could everyone give me a 6 in the chat if they follow up until right here? Okay. Now, F is our different form of our enzyme that's uniquely capable of binding to B. And whenever it binds to B, what it's going to do is it's going to take this methyl group off of its chin and put it onto our second substrate, B. And effectively, I'll just draw B is represented by a square. B is the square, therefore, the square with the methyl group is none other than Q, which is our second product. And as soon as our Pac-Man lost the methyl group, well, now it reverted back to its original state. Okay. Now, could everyone give me a 3 in the chat if they feel all right about this? And a E in the chat if you feel not all right. Okay. So what I'm going to do is I'm going to redo this ping pong model. We have a couple of different players. E is our enzyme. F is also our enzyme, but with a methyl group. Okay. A is our first substrate, which is represented by a circle with a methyl group. R is represented by, which is our first product, just represented by that colored in circle. B is our second substrate, which is a black box. And then Q is our black box with a methyl group on it. So original enzyme, modified form of our enzyme. First substrate, first product. Second substrate, second product. At the end of the day, what's happening is our enzyme, E, is taking a methyl group off of our first substrate. It's holding on to it temporarily, and or it's holding on to it temporarily until it adds it to the second substrate. Now, to kind of see the flow of all of this, we have our free enzyme. E. That binds to A. From there, we get the EA complex. The EA complex represents our enzyme bound to our first substrate. Whenever those two things come together, a reaction takes place. The methyl group, so when we get E and A together, the methyl group is transferred from A onto E. And whenever the methyl group is added onto E right here, the result is F. Now, when we remove, the only source of that methyl group is coming from A. And as soon as that methyl group is removed from A, then R is what we're looking at. So FR. So the FR complex is our modified form of our enzyme with our first product, where R is then released by F. Could everyone give me a V in the chat if they get it up until here, or a I in the chat if, no, I'm still mixed up. Sorry, I meant V as in Virginia, but B as in Virginia also works. Okay. So as soon as FR complex releases R, the result is F, our modified form of our enzyme. I'm going to change colors for the purposes of organization. That F complex, or that F modified form of our enzyme then binds to B. Okay, so the result of that is FB. At this point, we have F, which is our methylated form of our enzyme. What we are going to do is have F and B exist together. F is going to transfer the methyl group onto B. What is the methylated form of B? None other than Q. What is the demethylated form of F? None other than E. So this reaction is the transfer of a methyl group from F onto B, which results in E and Q. Our second product, Q, is then released. And our enzyme, E, has reverted to its original state. So this is a ping pong model because one substrate has to be converted into the first product before a second substrate can bind. So there's lots of important information about this and lots of things to understand about this. First of all, our enzyme, E, in its original state, cannot bind B. Likewise, our modified form of F cannot bind to A. It can only bind to the substrate, or our second substrate, B. Now, there is a lot going on here, but this is just a way of visualizing reaction models. Now, this is all summarized and applied with specific reactions down here, where we have a ping pong catalysis where our enzyme undergoes minor changes to form a different version of itself. For today, what I want you guys to do is I want you to have the conceptual understanding of this. After you feel comfortable with the conceptual understanding of these three models, then I would go ahead and take a look at these. I'll ask you some questions about these different models, but for today, in this class period, I want you to feel comfortable with random, ordered, or ping pong. Now, with that, it's 4:45. I want to do a little bit of stuff. Yeah, I'll get a couple more slides. So now we're going to switch gears back to a conversation about hemoglobin. Now, one of the important things about hemoglobin is hemoglobin can bind to O2 in the presence of that heme with that iron ion. I said that, hey, O2 doesn't change. It doesn't. But the complex, the oxygenated form of hemoglobin is different than the deoxygenated form. Now, this is something that is different with respect to the light spectrum. So within the visible light spectrum and the UV light spectrum, you have a little bit of a difference. And this principle is really a good way of explaining how your watch works. So my watch is a Garmin 945, and it tells me that today I've walked 3,970 steps. Mondays and Wednesdays are my low step days. Um, what this has within it is an instrument known as a pulse ox. Okay, got it. Okay, good. I'm seeing it makes sense. A pulse oximeter. And what that is going to do is it's going to tell me basically what my O2 saturation is. And that O2 saturation is going to be looking at the relative amounts of oxygenated and deoxygenated hemoglobin in my bloodstream. It's going to compare those two things and it's going to say, well, based on these two numbers, you have approximately this much, or this is how saturated your blood is with oxygen. Now, actually, you know what? I'm going to take a stop right there because what I've got is I've got the closer questions. I want to give you time to answer those closer questions. One thing, though, about the closer questions is that, let's see, the second one is getting to something that we are going to tackle on Monday. For the time being, though, I want you to understand that a functional protein might change whenever it binds to its ligand. I want you to understand this terminology. I want you to understand the difference between a substrate and a ligand and when they are applied. Now, in the meantime, I've got a discussion for today up. I'm going to get started to make sure that all of your grades are up to date, hopefully before the end of this weekend. Um, but this is where we're going to stop right now. We'll pick up the rest of it on Monday, and we will go into conversations about enzymes and protein functions. In the meantime, though, if you guys got any questions, please don't hesitate to ask. Otherwise, I hope you have a great weekend. If you need office hours tomorrow or Friday, email me with the time and I'll get back to you to see if that works for me. Hope that it will. But thanks very much for coming to class. My question's on the Word doc. Yes, I will get back to that question that's on the Word document. I might send that as an email because it has not crashed yet. But thank you all very much for coming to class. And I will stop my recording right now.
Thanks, Dr. Gray. Bye.
Absolutely. Thank you.